Welcome to the tour of a lifetime. We're going to be taking an excellent adventure through the inner workings of an animal cell. Along the way, we will stop and talk about each wonderful organelle and its function. Now hop inside the magic shrinking El Camino and we'll begin our excellent adventure. In order to see the insides of this animal cell, we'll have to pass through the cell membrane. This membrane acts as a selectively permeable barrier and is made of a phospholipid bilayer which has a hydrophilic polar head and a hydrophobic core. This is really interesting because this is where passive and active transport takes place and is where certain materials enter the cell, like we're doing right now. What we're looking at now is the cell nucleus. This is really super cool because this is where most of the genes that control the eukaryotic cell are located. It acts as the brain of the cell and coordinates what the cell does.
What's that thing in the middle of the nucleus?
That is the nucleolus, and it creates ribosomes, which we'll see more of later on. Nucleolus actually means little nucleus, interestingly enough. That genetic material in the nucleus is called chromatin. It consists mostly of proteins and DNA and compresses to form chromosomes, but only during mitosis. As we move out of the nucleus, we pass the nuclear envelope, which is what separates the contents of the nucleus and the cytoplasm, and is perforated with many pores. Now we are fully outside of the nucleus and floating about in cytoplasm, which I will talk about momentarily. Now we'll look at the endoplasmic reticulum, or ER for short. It channels proteins into some transport vesicles, which we'll also see later, and attaches carbohydrates to some proteins. Those little doodads attached to the endoplasmic reticulum are called ribosomes. They act as little protein factories. These proteins can be used inside or outside of the cell. These ribosomes are attached to the rough endoplasmic reticulum and give it a rough texture, which is where the rough ER gets its name, although not all ribosomes that exist in a cell are bound to the rough ER. Another part of the endoplasmic reticulum, but not part of the rough ER, is the smooth ER. As you can see, there are no ribosomes attached to this part. This part of the endoplasmic reticulum is responsible for lipid and fat production within the cell. Now that we have the ER behind us, we can now talk about this gunk we're all suspended in. It's called cytoplasm and is the entire region between the nucleus and the cell membrane. This means that the cytoplasm houses all of the organelles inside of the cell. I'm sure you guys are all eyeing that thing right there. I'm glad you're all so curious. That is the mitochondria. This is what produces ATP within the cell, and uses various sugars, fats, and other fuels to produce that ATP. This ATP is what makes active transport possible within the cell membrane. Those little floating thingies are called vesicles. There are two types of vesicles, secretory and transport. Secretory vesicles carry products modified by the Golgi apparatus to the cell membrane and secretes them outside of the cell. The transport vesicles carry ER products to the Golgi apparatus, which we'll talk about next. The Golgi apparatus receives ER products from transport vesicles and modifies those ER products in order to be secreted by secretory vesicles. The Golgi apparatus also creates these secretory vesicles and creates lysosomes as well. Speaking of lysosomes, here they are right now. Lysosomes are a sack of hydrolytic enzymes and are used to digest foods, other molecules, and worn out cell parts. Now don't get too close, children, or you'll be digested by that lysosome too. It's also worth noting that this particular organelle is not present in plant cells. You guys really lucked out. This is called a vacuole. The vacuole is a membrane-bound sac that are larger than vesicles. These store materials such as food and water for use in the cell.
But teacher, why is it so small?
Good question. Vacuoles in animal cells are actually much smaller than they are in plant cells. Vacuoles in plant cells are actually used for structural purposes, unlike vacuoles in animal cells. This is a centriole structure. This is found in animal cells and are used in cell division. They are made up of microtubules and microfilaments. These are the microtubules and microfilaments. Microtubules are hollow tubes that consist of 13 columns of tubulin molecules. These are responsible for maintenance of cell shape and chromosome movements. They are 25 nanometers. Microfilaments are two intertwined strands of actin and are only 7 nanometers. These are also responsible for cell shape and also muscle contraction. Well, that concludes our little tour. I hope you guys had a wonderful and interesting experience. We're going to follow that secretory vesicle out of here.